Welcome to Science Sundays. I'm really impressed at such a beautiful, sunny afternoon that you're all here to learn about ocean viruses. Science Sundays is a public lecture series. It's a joint effort of 8 centers within the College of Arts and Sciences and the Office of Research. We hope you've been enjoying this year's presentations. We've got a bunch of really good ones that we're lining up for next season. Before we go any further, please also silence your cell phones. I appreciate it. So my name is Dan Schoenberg. I'm the director of the Center for RNA Biology. And for those of you who hear about nucleic acids and medicine things, you hear about DNA a lot. You don't hear that much about RNA, but we actually live in an RNA world. RNA was the primordial nucleic acid. Some people believe life arose from RNA. All the information that's encoded in your DNA, you can think of it like a hard drive, gets transcribed into RNA, and RNA becomes the functional molecule that carries information from the nucleus into the cytoplasm to direct protein synthesis. It actually catalyzes some of these reactions itself. It's a regulatory molecule. And it's also, and for many viruses, it is the molecule that carries genetic information on the virus. Examples are hepatitis C virus or HIV that causes AIDS. And so it's fitting that today's talk is from Matt Sullivan. So Matt is an associate professor in the Department of Microbiology and Civil, Environmental, and Geodetic Engineering. He's co-founder of the University of Arizona Ecosystems Genomics Institute. His research aims to quantitatively explore the roles of microbial viruses in ocean geochemical cycling, thawing permafrost, and humans. As such, he's helped lay the foundation for modern quantitative viral ecology. His work has significantly expanded our understanding of the global virosphere through illuminating viral dark matter. I think we'll hear a lot about that today. And he's pioneered numerous experimental and informatics approaches to link and explore virus-host interactions. Matt's received numerous awards and recognition. He was a Fulbright Scholar. He's the virus coordinator for the Global Terra Oceans and Malaspina research expeditions, which I think you're seeing some movies of here. He's a Gordon and Betty Moore Foundation investigator, a Kavli Frontiers Science Fellow, and a Beckman Scholars undergraduate research mentor. Before I turn the podium over to Matt, there's a couple points I want to make. First, when you come, please hold your questions till the end, and then people under 18 get first crack at the questions. So please let them ask first. And now please join me in welcoming Matt Sullivan, who will speak on understanding ocean viruses, may just save the earth and help cure your next ailment. Matt?
Thank you. Thanks for coming. This is a research cruise. So any of you who are not seasick, I'm impressed. I get very seasick. This is on the Tar Oceans, running around in a place where the winds do blow consistently. So it's not always this bad. Many times you get beautiful ocean waters. But the next time you hear someone say they're going on a research cruise, don't look at them and say nice vacation. They may actually be working very hard. So I want to talk to you today about viruses, in particular viruses of microbes. I don't know really much at all about viruses that infect you, but I know a lot about viruses that infect the microbes that dominate your body. I know a lot about the viruses that infect the microbes that dominate soils and the oceans. And the way I want us to think about the viruses I'm going to introduce for the first half of this talk is that these are ocean viruses, but they're really important as we learn how to study them and think about a roadmap for studying viruses of any microbe. Again, microbes that are in your body, microbes that are in soils, microbes that are important in industry. So we're really going to structure the talk around these 3 questions. Why study viruses and microbes? What do we know about viruses and microbes, and in particular I'll focus on the oceans? And how does this help us in humans? So a virus is only as interesting as its host, and many of you may recognize that microbes are important for things we care about, like beer and wine, bread. They're important for bioenergy. They may be a way to get us towards sustainable energy futures. And if you've had a radio on in the last couple of years, you've heard all about microbes in your body. You've heard that for years and years, decades, that microbes cause pathogenicity and disease. But we're now learning that there's also microbes that live with us in a more normal manner, and that those microbes may actually manipulate things like your diet help you digest certain kinds of foods that you wouldn't normally be able to digest. We're also learning these microbes may actually control your behavior. They produce compounds which can actually speak to your brain and cause you to want to do things. They also can control whether you're obese or lean. Now, microbes are doing this in the context of a human body, and so I'm going to suggest that that human body is, to me, just another ecosystem. It's another interplay between some large organism and a bunch of little organisms and the communication and language that they have between them. So those microbial roles I'm hoping you've heard a bit about. You may not know that on Earth, much of the redox chemistry that manipulates the kinds of energy and nutrient transitions which run planet Earth are all in this red box. And that redox chemistry is predominantly driven by enzymes, little pieces of machinery in a microbe. So these microbes are not only important in your body for industrial purposes, to make breads, wines, and bioenergy, these microbes are also out there in the wild doing a lot of important things running planet Earth. To give you some examples that might hit close to home even further, if you had a bowl of cereal this morning or some bread on a sandwich for lunch, you probably could thank the microbes for the nutrients in the soil that fed the plant that ultimately fed you. If any of you have been breathing since you walked in the door, every other breath, the oxygen in that breath comes from marine microbes, microbes in the oceans. This is really profound. You can actually see these microbial blooms from space. This is a picture, zoomed in now, of Earth. And this microbe is called a coccolithophore. It's a photosynthetic cell that has these very cool calcified shields on its body. These coccolithophores, these microbes, are helping to control carbon dioxide. Why should we care about carbon dioxide? This is a plot of the last 400,000 years, though we have data for the past 650,000 years. Basically, what we've learned is that, yes, carbon dioxide in the atmosphere cycles. There is a natural cyclical progression of carbon dioxide. And for 650,000 years, it's never gone above 300 parts per million. You can see that around the time of the Industrial Revolution, when we started producing carbon dioxide in ways that were pretty advanced for humans, you can see that it jumps way above 300 parts per million. In fact, successfully last year, we broke the 400 part per million barrier. Now again, you need to thank the oceans, not just for that oxygen that you breathe, but also because they're helping with this carbon dioxide problem. Many of you are probably aware that when we have too much carbon dioxide in the atmosphere, it's a greenhouse gas and it can cause climate change, warming, instability, more extreme weather. The people in California right now certainly know about that. Here we're showing the actual quantitative measurements. You see that burning forests, we release about 2 gigatons per year of carbon dioxide. And through fossil fuel burning, we release about 5 gigatons per year. Now you are all doing the math and saying 2 plus 5 is 7. But above that, I see that the atmosphere has only increased 4 gigatons per year. Well, what's happening? It turns out the oceans are helping us. They're taking down about half of that carbon dioxide through mass action. And then when that carbon dioxide, which just diffuses into the ocean, gets into the ocean, it's part of a buffering capacity of the ocean. It actually is part of a pH changing capacity. So at least for now, the oceans are capable of buffering half of that carbon dioxide that we put into the atmosphere. You'll also see that at the bottom I flagged that in the oceans, about 3 gigatons per year, that which is the sink, sinks out of the oceans, out of the surface oceans to the deep ocean. And that's good for us because that means maybe we have a few hundred or 1,000 years to geoengineer a solution to our large carbon dioxide atmosphere problems. And the other number I've decided to highlight was viral lysis. The biggest flux of carbon in the ocean is due to viral lysis. So that sinking number is going to become important later, but for now we're going to focus on carbon dioxide and pH. Now if the oceans have sucked down a lot of carbon, right, 3 gigatons per year, you can't possibly have changed the way that the ocean looks, right? So let's go to the middle of the Pacific Ocean and the Atlantic Ocean, the 2 largest oceans on our planet. And there's actually been long-term ecological research sites that have been studying, making measurements there for decades. One's called HOT for the Hawaii Ocean Time Series, and one's called BATS, for the Bermuda Ocean Time Series. And you can see a dot in the middle of the ocean there where each of these is located. This is a plot of the story at Bermuda, and you can see there's annual cyclicity in both the measurement on the top, which is the amount of carbon dioxide in the water, and the measurement on the bottom, which is the pH of the water. So there is a seasonality, which actually comes with those phytoplankton, or those photosynthetic cells I mentioned earlier, seasonally blooming or not. But you can see the trend line is up. In fact, we've managed to decrease the pH of the oceans by adding carbon dioxide to the atmosphere and the ocean sucking it down as part of its buffering capacity. Now that's the Atlantic Ocean. The same is true in Hawaii. This is in the Pacific Ocean. Over a couple of decades, just the last few decades, we've managed to acidify the ocean. So you've probably heard of ocean acidification. It comes up in the news now and then, and there's big worry that that'll impact a lot of calcified organisms in the oceans. I'll just suggest that maybe viruses play some role in this again later. So that's hopefully why you should care about studying viruses in the sea. They may play some role in this big carbon pump that right now is helping us have the atmospheric carbon dioxide problem that humans have produced. So what do we actually know about these viruses? We actually didn't think there were viruses in the ocean for many, many, many years. Someone went out with an E. coli, a common bacterium that you would use in a laboratory, but not a very common bacterium in the ocean. They used it as a host to isolate viruses, and they didn't get any. And then another group did this. They just collected seawater and put it on a filter, and they stained it, in this case with a DNA stain, and the small dots are viruses, and the big dots are cells. And hopefully you can see that there's a lot of small dots there. There's a lot of big dots. We learned that the small dots are about 10 times more abundant than the big dots. So not only are there viruses there, but they're more abundant than the cells, the microbes, which are the base of the ocean. So let's talk numbers. Next time you swallow a mouthful of seawater, 50 million viruses. Don't worry, they don't infect you. They don't even infect your microbes. They infect ocean microbes, and they're so adapted to affecting those ocean microbes, they're not going to hurt you unless you're swimming outside a sewage outfall. And just to give you a few big ecological numbers, and these are not well-constrained numbers, but we'll use them as a starting point. About 1 out of 3 cells in the ocean are killed per day. So the oceans, or the viruses, play an important role in mortality. About 10 to the 29th genes are moved by viruses per day. What does that mean? That means that a virus can infect a cell, and it can accidentally pick up part of that DNA and infect another cell and move that DNA. This has a name called transduction in genetics. And so viruses are moving 10 to the 29th genes planet-wide per day. That's a lot of ways that viruses help change their host cells. So they can actually help host cells do different things. One good example might be antibiotic resistance. We hear a lot about antibiotic resistance moving between hosts and spreading easily. One way that could happen is through viruses. Another reason you might or should care about viruses and ocean viruses and other environmental viruses is that they encode toxins. In fact, if you ever thought cholera toxin, Shiga toxin, Bordetella toxin, pertussis toxin, you thought these were microbes that cause them. It's actually a virus integrated in the microbial genome that has the toxin. So the virus has created a relationship with that cell by keeping that toxin in its own genome. So the viruses encode many of these bacterial toxins. And then this is only important in that it's hard to work with these viruses. Less than 1% can be cultured. That means we don't sort of know the magic formula yet to grow their hosts, let alone to get their viruses. And so much of the inferences that we're going to make are going to be using sequencing technologies. So what do we know about these viruses from sort of latest work? Those numbers were summarizing a couple of decades of work, actually. So I was fortunate to be the virus coordinator on a boat called the Tara Oceans. And this is the Tara. And this was a lot of interesting scientists who wanted to study viruses to fish larvae, all these organisms as planet-wide as we could get that boat. And the basic idea is that we have this sailboat that's going to sail around the world and it's going to stop at these red dots and throw that CTD overboard, which has a bunch of bottles in it. Each of those red dots is 24 to 48 hours of sampling. So again, the research cruise, where you feel like someone is on vacation, remember that every couple of days they're stopping to work for 2 days. And then the other point I want to make is that to do this, and you'll see a little more what the boat looked like, we had to develop some really innovative new ways to quantitatively and systematically sample. We wanted a picture of viruses worldwide, which means we had to sample them consistently. And we wanted to do it quantitatively, because at the time, the methods that we had were very poor at representing what was actually in nature. This is another shot of the Tara. She's absolutely beautiful under sail. She is a sailboat, which is a very busy sailboat. Again, we had 7 scientists on board at any time, and so those 24 to 48-hour sampling expeditions, you could imagine, are challenging. This is the CTD, the round bed at the bottom, and then that house that you see in the back is what was called the filter house, or the wet lab. If we take a look inside that wet lab, you had 3 people who would essentially stand back to back to back, doing a lot of filtration. If you go on a research cruise, you're not doing too much that's glamorous, you're really filtering water. This is what filters look like. They were stacked all the way to the ceiling. A lot of stuff that got swapped in and out, highly optimized for space and time and sampling efficiency. In addition to the wet lab, we also had what we called the dry lab, and if you needed some money, this was the place to rob. There was millions of dollars of equipment in this room under the boat. I can't believe that we brought some of this stuff out to sea. It's hard to imagine the instrumentation people ever imagining them on a boat. But there's lots of different equipment here to allow us to make a lot of neat measurements to contextualize the kind of science that we wanted to do. So that was what it's like to be on this boat and how to sample and the cramped conditions, and you're on it for 1 month at a time. What have we learned? So the first thing we learned is that, well, viruses are mostly unknown, as I hinted to earlier. We could look at thousands and thousands of viral populations, or we now call them species, and less than 1% had we seen before. But at least now we've got a genome, and we'll get back to that. I don't like looking at less than 1%. You go outside, you look under the street lamp, you can only see this tiny little bit. That means you're missing a lot, right? During the day, if you were to open this up, there's a lot of beautiful things to look at. So we wanted to illuminate this dark matter. Since Dan used those words earlier, I'll make sure to follow up. We were interested in exploring viral dark matter, or that viral unknown sequence space. And so this is a pictorial representation of what was known to all of science before we started. So there were 2,000 viral genomes approximately. And so each of these pictures of a virus represents 1,000 genomes. And after we were done sailing the high seas, we now know about 30,000. So we'd augmented existing viral genome databases 15-fold. So that's a big help. The next thing we did is, it turns out viruses don't actually have a barcode gene. If you hear anyone who goes out and says, I'm going to study diversity, or try to ask what kind of microbes are there, they're usually using a barcode gene. Turns out it's an RNA gene, the 16S RNA gene, because it's so important to those cells that it's conserved across all of life. We don't have a gene, even a single gene, that's shared across viruses, so there's no barcode gene. So we had to develop a genome and network-based taxonomy just to be able to say who these viruses are. So we collected, it turned out in this first cruise, added to the second cruise, about 15,000 different viral genomes. And in these 15,000 viral genomes, we had to figure out a way to relate them. And so we developed a network-based analytic. And this is powerful because it turns out it's going to be the new way forward for classifying any new viruses and microbes that get examined in any environment. And then this one shocked me. We're sailing around the world's oceans. You know, you saw how spaced apart those sampling sites were, even though it was 3 years of intense sampling. And we started to actually saturate the viral diversity. So this means that every time we collect a new sample on the x-axis, and we add the number of new types, or viral clusters, I've said viral clusters here, we're not getting any new ones, because you see that curve starts to level out up here. That means that we've saturated virus diversity in the ocean. So this is good. It means we've got a near complete catalog with which to start to study these viruses. Okay, so let's study them. So we did, again, sail around the world. We had all these different stations. We were interested in each station because of the oceanographic features that were there, different aspects of the ocean that we wanted to try to capture and study. And each of these open circles is a station, one of those 24 to 48-hour stations. And then you can see the cruise track moving from Europe through the Med Sea, around and about Africa, and then around and about South America and into the Pacific. And what we learned, using these genomes as essentially genetic tracking devices, we could actually follow the flow of these viruses. And the arrows that are colored from this previous slide are the direction which those viral populations seem to go. And I've just layered on the major ocean currents. So before we started, we probably could have said this: viruses ride ocean currents. Viruses don't swim. They're not going against the currents. They're going to be passive. But I'm just setting up that we can use these genomes here as a genetic tracker. And we're going to come back to this. So what did we learn? We learned that a lot of viral sequence space is unknown, and that we've now mapped most of it. We've got nearly a complete catalog, about 30,000 viruses that we have genomes for. So the second thing is we learned that viruses are impacting this carbon cycling that I started with. So viruses are doing that actually in a big way. So if we go back to this rather simplistic view of what we learned, that viruses ride the ocean currents, what I didn't tell you with the X's is that wherever there's an X, viruses were sinking. Now, viruses don't sink. They have a low Reynolds number. If you put a feather in a glass of water, it doesn't sink. Viruses should not sink. So what's going on? This was what we thought before this work. We thought viruses would infect bacteria or these photosynthetic cells called phytoplankton. They would lyse those cells and produce small organic matter, small pieces of cells. And those small pieces of cells would actually just feed more growth in the small cells. And so this was called the viral shunt. This was suggested that viruses basically keep carbon small. So that would mean in the surface ocean, all the cells that were lysed would be turned into small bits of organic matter that could be reused by the smaller cells. We would never get fish, for example. But what we're seeing is not consistent with that, and so we said, well, let's assume that paradigm that we've operated under for 2 decades is wrong. What else could be happening? And so we thought, well, these small bits of organic matter, it's a pretty local process. When a virus blows up a cell, it's happening right there at this microscale. What if that local process allowed for aggregations or sticky stuff to form? Or alternatively, what if virus-infected cells were better food for grazers? We could imagine viruses, through either of those mechanisms, producing aggregates. And those aggregates might actually sink out of the water column as export. Well, it's an interesting hypothesis, but how do you test something like that? So this isn't the complete test, but this is a very interesting set of information. So this is going to sound like a joke. 2 oceanographers and 2 theoreticians walk into a bar. So this is a study about genes to ecosystems modeling. Now, in my world, we've long sought, for at least the last decade and a half, to try to take sequence data and predict something about an ecosystem, or about your body, or about farming. And that had been a challenge, and nobody had done it. And so what was interesting here is these 2 theoreticians talked a lot with the 2 oceanographers, and then you can see there's about 50 other names there. A lot of us contributed to this, as well as that Tara Oceans data from viruses to fish larvae. And we just said, well, let's do some statistical and modeling-based approaches to ask, could we make some predictions from the things we've measured? And so here, we'd sailed around the world to look at carbon flux in addition to a bunch of other things. And so what's plotted here is the carbon flux measurements, where you've got, again, these white dots are the 24 to 48-hour stops. Interpolate between dots the carbon flux through the water column, and this was estimated from actually underwater cameras that we had, a very fancy underwater camera that went down with the CTD. And so when you plugged in all of the organismal data or all of the molecular gene genetic data, what you learned is that the best predictor of carbon export in the global oceans was viruses. And actually, by far, it was viruses. They could predict 89% of the variability of carbon export. And in ecology or studying anything in nature, if you predict 89% of anything, go ahead, write that paper up, it's done. It's a theory, it's done. So we were very excited about that. And what I think this is a first indication that here we've mapped in the global oceans these viruses, and they are the major predictors of carbon flux. Now, some of you may not care about carbon flux, so I'm just going to emphasize this other point, that the modeling approach that they did is actually quite generalizable. And so if you, in your any ecosystem, your gut, your lung, soils, if you were mapping organisms and you had something you cared about, say a disease or the ability of plants to grow, you could try to predict that, use the same modeling approach, and get it down to ask the same question. Which organisms or even which genes are driving the thing you care about? And so this is a new and emergent approach that just came out last year to be able to predict key determinants of disease or soil health or product yields in a bioreactor case. So a very powerful new analytic. So that's the oceans, and that's what I normally study and do every day. But part of my move here to Ohio State was I was beginning to realize that, first of all, I get the question, why do taxpayers pay for that? An awful lot. And I also, myself, wanted to try to see if some of this work we were doing was generalizable. And so I wanted to begin to say, how could we try to help humans? How could we really try to make a difference using these ocean toolkit in humans? So this is how we can help. First, I just alluded very briefly to this. We've got the ability to do quantitative surveys now. So what that means is that we can take a sample and concentrate it, purify it, amplify the very, very, very few nucleic acids we get in a way that's quantitative. And this is a powerful approach when you want to do things like predictive modeling. And as also alluded in the oceans work, we had to develop a network-based analytic to even know what to call those viruses. In the oceans work, we also had to develop the ecological modeling, the genes to ecosystems, predictive modeling, to be able to make predictions based on this sequence data. So I usually say, we now know how to count, and we now know how to make predictions. And that's a powerful thing. The other thing is, we're rapidly illuminating that viral dark matter. So I mentioned we took, all of science had 2,000 viral genomes, and in just a couple of years in the oceans, we were able to augment that from 2,000 to 30,000, and now we've got equivalently large contributions coming from soils and humans. So it's a very quick and changing landscape to allow us to just know what organisms to study. So I'm going to just tell you a little bit now in the human realm about how these, I'm going to call them ecology advances from the oceans, help us think about that human ecosystem. So this is the ecology of viruses in humans, and we're going to focus on two aspects. First, in the gut, and our particular study was looking at autistic children. And the second is in the lung, and we were interested in how smoking impacts your body. I'll tell you that we have some answers on the first, and on the second one, I won't have any answers, but I'll lay the framework for you. So what happens in the gut, and I have to admit, this paper came out a few weeks ago, and I've never gotten so many really wonderful emails from people who clearly are passionate about their children who have autism and want me to suggest that what we've done can solve their problem right away. So I'm going to start with the caveat that this was basically a bootlegged project. It had no money, and so the scale is we studied 18 autistic children and 20 neurotypic children, so that's a low sample size. And the other aspect is that many of the autistic measurements in the clinic are frankly subjective. Okay, so questionnaire-based stuff. And so if there's an improvement on the autistic side, I just want to flag this was an open label study, meaning that people knew they were being treated or not. And so we're really excited about what this first pilot study did. But before I get too many folks coming up saying, I want to sign up, I just want to flag that first study. Okay, so what's the idea? In autistic children, apparently there's a connection between GI symptoms, and basically that once you have these GI symptoms, that immediately implicates the gut. And so people before my time had been suggesting that maybe gut bacteria were important in causing autism. And in particular, you could have a reduction in diversity of these gut bacteria, and you might also have a reduction in fermenting gut bacteria in particular, and this has been observed in the gut. So there's correlations suggesting there may be a connection in autistic children in their severity of symptoms having to do with GI problems. Well, as I've tried to suggest, viruses play a role in that ecosystem as well. Viruses are in the gut. In fact, there's quite a number of them, and they play lots of different roles, and so maybe they could be important here. So we worked with two really good collaborators in Arizona and just approached autistic treatment with something called fecal matter transplant, and in particular we did a long version of it. So basically for 10 weeks they were taking this fecal matter transplant, and the idea was to replace the autistic gut's microbes with donor microbes from a healthy individual. And so what did we find? Well, we did find significantly improved by 80% GI symptoms, and that's a real number. That's a very quantifiable number. And 16 of the 18 children, by these more subjective surveys, their autism symptoms had improved. So that's really positive and exciting and a great first step. And again, because of these quantitative survey tools that we now actually have a good handle on a number of bacteria and viral populations or viral species which correlate with these improvements. And so a next step for this kind of work, if we can get to a next stage clinical trial, would be to obviously increase the size and power of the study, but also try to think about specifically looking at some of these groups that came out as strong corollaries. So this is a very simple, from my perspective, scientific design that's solely looking at ecological changes in the body over time. And we finally have the tools to do this both at the bacterial level and the viral level. And then in the smoker's lung, we were interested in the fact that it seems like smoke should be some sort of stress and should change your microbial community. And actually the literature suggests that non-smokers and healthy smokers don't have a significant difference in their bacterial loads, but we suggest that, or we hope that there might be something interesting on the viral front that could be important. And we don't have an answer to that yet. We've got the first round of data coming in, but I'll tell you that it does look like we're guessing right in that the viruses do change between the smoker's lung, and we think that could be sort of a canary in the coal mine, a first indicator of changes to come. And so we're interested in trying to design appropriate longitudinal studies. And again, this is solely using an ecological approach. And another viral ecologist named Forrest Rower had done some really neat work, and he's been in the human system for about 6 years, though he's really driven by coral reef ecology and before that, open ocean ecology. And I thought this was a fascinating story, and so it's not mine, but I'd love to just share it with you. So the idea is, in this case, it was the coral mucus, but they found that actually it also happens in humans, in particular in the CF, the cystic fibrosis infected lung. And so what they found is that cells could produce mucus, and that mucus could actually bond with phages, and those phages end up sort of being part of the mucus layer. And then if you get a pathogen which comes along, it actually hits phage, and those phage are viruses of bacteria. Those phage are right there to infect that pathogen. And so essentially what they suggest is this is a symbiosis. This is a human or a coral producing mucus, probably for other reasons, but one reason is that it collects and allows phage or viruses to get in it. And these are not viruses of the coral or the human. These are viruses to kill pathogens. And so they've suggested this is a non-host derived immunity. And so you're using viruses in this way to try to get at immunity. So again, those are 3 examples of ways that ecological studies can really help us in the clinic. And the other way that I'm particularly excited about here is putting my engineering hat on and also sort of a reductionist microbiology hat. We're interested in mechanism. In particular, we've been interested in mechanism for how do bacteria resist viruses? How does resistance to viruses occur? And in the context here of what's called phage therapy, and we'll come to that. So for a while, we've been interested in how a virus could infect one cell, and that could lead to lysis. But if something happened to that cell and it changed just slightly, that could lead to no lysis. And this wasn't really all that well understood. And so we wanted to start to launch a bunch of different, what we call model systems, or virus host pairs that we can grow in the lab to understand what are the mechanisms by which this resistance occurs. Now, why should you care? Well, you've probably heard of superbugs, or these antibiotic-resistant bacterial pathogens. You probably have also heard of them as different names like MRSA, or C. diff, or Pseudomonas. These are infections in the hospitals which can be real big problems. You can go in for a arguably routine surgery and spend 45 days fighting a C. diff infection. And the reason is that these are bugs which have obtained antibiotic resistance gene cassettes, and so they're just really hard to kill by our traditional antibiotic methods. And so it's a major problem causing a lot of money and issues in hospitals. And so I think a reasonable solution is the 100-year-old idea of phage therapy. And this is pretty simple. Phages are viruses and bacteria. And you could imagine these viruses could be isolated such that they infect particularly that superbug and kill it. So what we're doing on that front is developing ways that we can pull down the right virus in a high throughput manner, make sure it doesn't have toxic genes, because I don't want a virus to spread Shiga toxin, cholera toxin, pertussis toxin, etc. And then make sure it doesn't kill the commensal microbes in your body that you actually want to keep around. And so what that involves is stuff we were already developing in the oceans. We were trying to, in high throughput manners, pull down viruses of hosts of interest and make sense of them, turn around and characterize them. And so we're trying to turn those ocean high throughput engineering-based approaches into tools for fighting superbugs. So I want to make sure you're clear that microbes are critical to life on Earth, and actually they're being mapped everywhere. In the human body, we've alluded to the fact that they're being mapped in your gut and your lung, on your skin, many other body parts, your mouth. This is being done to help combat disease. You saw that the Tara Oceans project was designed to go out and map these microbes throughout the world's oceans. There's also an Earth microbiome project, because why stop with the oceans if you could do the whole Earth? And my lab's goal really is we're interested in how viruses modulate these microbes in their outputs. So not only do they kill these microbes, they also alter the kinds of compounds they produce during infection. And so we study that at a mechanistic level as well. And then, as I alluded to in the phage therapy slide, I think we can use these viruses to actually tune microbial communities to desired states. So there's a lot of really smart people running really important studies to map these microbial communities. They're going to know, they're going to be able to tell us what microbes we want in our bodies, or in soils, or in a bioreactor. And I want to help us get to that point where we have those microbial communities tuned in such a way that we have a healthy state. So I think as viruses, as nanoscale entities with global scale impacts, and I hope you do too after this lecture, the contribution on the ocean side is that we've developed these quantitative sampling approaches, and that's critical for just making maps, just beginning to get a sense of whose they are. We've also used that quantitative approach to develop catalogs that are pretty nearly complete for the oceans, and so I think it'll be pretty reasonable to go ahead and get all the viruses in the human body, get all the viruses in soils. And I've suggested that to do or answer the questions we were interested in, we had to develop novel analytics, this genes to ecosystems modeling. And that genes to ecosystems modeling is valuable for making predictions. And in our case, we cared about carbon flux and learning that viruses were the major driver in carbon flux and that important role of the oceans in having our human-based impact of carbon dioxide. But this could be generalized to any microbial system, in the human, in the soils, in a bioreactor. I've also suggested and shown how we've started to take those ocean-based tools into humans, in particular in studying the autistic gut, human lung, and how we're starting to adapt that now for phage therapy. I'm just going to thank a few people from my lab who've been thanked along the way, but I'll leave their names up here as people who have really driven a lot of the work I specifically spoke about here. I want to thank two funding agencies, in particular the Joint Genome Institute and the Gordon and Betty Moore Foundation, who really took a flying leap with me, if you will, early on, and let me do a lot of high-risk and fun things. Thank all of you for listening, and be happy to entertain questions. Kids first. Kids first. Yeah, right in front.
I'm just wondering if do the garbage patches and the campaign specific affect any of the viruses and the bacteria?
Great question. So the question was, do the garbage patches in the ocean impact viruses or microbes or maybe even the phytoplankton in animals? So Tara, I didn't talk about it today. On the Tara Oceans project, we actually brought out special nets to collect plastic. So when you have a plastic bag from the grocery store and it ends up in the ocean over time and the UV light break it down into small things that are called microplastics. So microplastics through much of the oceans can be as abundant as the animals, the small animals in that water. There's two groups that are starting to study the microbes that live on that plastic, but those data aren't out yet. And there's no group studying viruses yet. So I'm sure that they're yet another resource for microbes to use in the oceans, but it's sort of a new frontier. It's being newly studied. Yeah. How far down did we get the sampling from? So I showed data from two global cruises. One was the Tara Oceans cruise, and that one was on the sailboat limited to mostly photoxone sampling and then about 1,000 meters depth, so into the dark ocean, but not near the bottom. And then the Malaspina ship from Spain was a real oceanographic research vessel, and that one we were able to go down to even 3,000, 4,000 meter depths. Yeah, so there's sort of two questions. In our autism study, were the viruses or microbes causing the improvement in autistic symptoms? And the second question is whether the fecal matter transplant comes with its own viruses in addition to just the microbes. So both of those, so the fecal matter transplant does come with viruses and microbes, and so you could be getting donor viruses. And so we did a longitudinal study out to 18 weeks to try to understand that dynamic a little bit. And even 8 weeks after the fecal matter transplant stopped, we saw engraftment. So we saw what looked like the recipient keeping the presumably beneficial donor viruses and bacteria. As far as a causative agent, we definitely don't know anything yet. What we see are that there's 3 genera of bacteria which are correlated with the improved symptoms, and there's a handful of viral populations which are correlated. And so we're interested to kind of think more deeply about causative mechanism, but yeah, it's a 2-week-old study. Yeah, so these are the GSRS clinical study, and there was 2 other clinical studies, and I'm not the MD, I'm a PhD, so the wrong doctor to ask that question of. But they saw improvements in basically sort of the problematic behavior in the classroom, so the teachers were surveyed. They saw improvements at, example, the dinner table at home, so the parents suggesting that the outbreaks and sort of frustration behavior were better. And I should know more some of the other behavior improvements, but you know, in this study, the director of the ASU Autism Institute, he's been the director for 20 years, was in tears when he showed us these data, because he's like, this is just so different than anything I've ever seen. So I'm nervous because it's out of my area of expertise, but you know, he's an expert and he was really impressed. And so I think we haven't done anything magical here, and a number of people in the world would predict that gut microbiota are critical to the autism phenotype, but it's you know, the next step will be starting to get at those causative efforts. Yeah.
So stepping back a little bit, it seems unlikely that we would learn so much about viruses through an oceanography research versus land-based or even body-based, because it's so available, land and body is so available. How did this come to be that you researched in the ocean?
Why did the oceans lead the way? Yeah. Well, I think they're easier to study. So I'm learning a lot about how hard it is to study anything in a human. You know, even to get fecal matter is a lot of work. So I think it. You know, I say that seriously. It turns out that Gordon and Betty Moore, I think, have a real vision and realization about a decade and a half ago about how important the oceans are to the Earth and the way it functions. And so they put a lot of money, to be honest, into scientists in that field to do specifically cutting-edge, innovative technical developments. And so I think a lot of it was, the money was there to do risky science. And I think the challenge when you write proposals to more traditional funding agencies is, if you can't already do it, you're not going to get funded to do it. And so I think that's why we're seeing a little bit of the leading from the oceans on that front. There was a high risk, high reward pot of money. Yeah. Yeah, so the question is really, is there a strong biogeography of virus populations? Do you see specific viruses in one place and not another? We haven't done a lot of work. There's almost no data on river gradients, so from a river to open ocean data set. But in the oceans, what we observed is that at any given sample, you had really all the diversity. If you sequence deep enough, you'd see all the populations we'd see globally. And what changed a lot was the abundances. So you'd have, in one environment, perhaps selection for microbe A and its viruses, in another environment, selection for microbe B and its viruses. So there's a decades-old idea of everything is everywhere and the environment selects. And so the data that we collected and our observations from it are consistent with that. Why the Department of Energy is a funder? Yeah, so the Joint Genome Institute for a long time has, I think, recognized the value of technical innovation as well. And so I've been lucky to excite them about the fact that viruses are probably important in many ecosystems that are important for bioenergy, for bioremediation, and other major mission elements of DOE. And again, they care about high risk, high reward innovation. So I think they just saw that there were some good ideas that could help. And in fact, I was just at a grantees meeting last week and speaking with the program directors. They're ready to shift some of the more mainstream sort of traditional DOE programs you'd think of towards the kind of toolkit that we'd developed and that they'd help support develop. So I think it was a good call. They can now study viruses in bioreactors that are critical for bioenergy, for example. But it is, again, that mentality of high risk, high reward, and the value of alternative systems. I'll say that it was really hard to get that funding until we sort of proved we could do it, kind of well, in that we were good at the high risk, high reward game. But once they sort of recognized that we had some good ideas now and then, I think they're ready. Yep. Question here. Yeah, so the two questions are there many parallels between viruses in the oceans and on land, and the ratio of retro versus DNA viruses, retro being some sort of RNA viruses. So the dirty secret that I didn't mention here is all of our toolkit is quantitative for DNA viruses, and in fact, at the time it was double-stranded DNA viruses. We now have quantitative single-stranded and double-stranded DNA viral approaches. And we're working on quantitative RNA approaches. Currently, there's no way to relatively examine RNA versus DNA viruses unless the center has a good idea for an RNA stain that's specific just for RNA and bright enough to see in a tiny viral particle in the background of a field sample. So we don't know the relative ratio. There's one study from the oceans that suggests that RNA viruses are between 17 and 75% of the total viral community, but there's a lot of issues with that data, and it's one sample.
One more question.
Up front here.
Congratulations on the discovery of some new virus. I'm curious if that discovery was just unexpected, if there was any expectation to be so successful, and also whether there's any expectation that it might happen again in some of these areas.
The question is, were we surprised by that diversity, and would I expect others to meet that same diversity in maybe new environments? Yeah, I don't think I was surprised that there was a lot of new diversity to be discovered. I was surprised that we could saturate that sampling. Going into it, there was a paradigm that viruses were the largest genetic reservoir on the planet. And in fact, we were dead wrong. It's the opposite. Viruses are probably the least genetically diverse of all organisms. There's a lot of data by a couple orders of magnitude. And so I'm excited because I like finite problems. I like to bound the problem. And certainly I think new environments are going to have new viruses, at least ones that aren't connected to the ocean. So the human, the soils, we're already seeing a lot of novelty, but again, we're starting to saturate in both those environments. And so I think it's a fun game to keep going at and get the catalogs. And what's exciting is now that you have the catalog, you can do some real science. It helps anyone do this science in a way that, that is powerful. So.
That, please join me in thanking Matt.